All right, so we've talked about how we can synthesize and replicate DNA. The next thing we're going to talk about is how to actually get that information from our DNA. So just like the information stored in your textbook is completely useless until it is read and put to use, the information coded in DNA is functionally worthless until that information is read and acted upon. Genetic information is put into use by the production of proteins. In other words, gene expression is simply turning that genetic information into proteins. Genetic information is going to be expressed through protein synthesis, the expression of genetic information into the structure and function of DNA. DNA is going to be read and then translated into RNA. RNA then controls the primary structure of the protein. Does anybody remember what the primary structure of a protein is? Yeah, it's the sequence of amino acids. The sequence of amino acids determines the overall shape and structure of the protein, and the sequence of amino acids is determined by the sequence of DNA. So RNA is our carrier of information. Like DNA, RNA is composed of nucleotides. It is a polymer composed of nucleotides, but just like DNA, RNA nucleotides contain a 5-carbon sugar called ribose instead of deoxyribose. That's where the R comes from instead of the D for DNA. The RNA molecule nucleotides, come in 4 types: adenine, guanine, cytosine, and uracil. Uracil being the main difference between RNA and DNA. In DNA, we have adenine, guanine, thymine, and cytosine, and in RNA, we have adenine, guanine, uracil, and cytosine. RNA is also going to be a single-stranded molecule as opposed to DNA, which is a double-stranded molecule. RNA is not going to be wound into a helix. There's no need. It's usually a very short sequence, and it is single-stranded. DNA, on the other hand, is wound around in a helical shape. Now, there are 3 major classes of RNA that are involved in the protein synthesis. The first is messenger RNA. Messenger RNA is like it says, it copies down the genetic information from DNA and it takes it out of the nucleus to the ribosomes for the synthesis of proteins. It's kind of like when you take notes down in class. You're just going to take down what you need to know, and then you're going to walk out of the classroom and take that with you. So this process of copying down that information is going to be called transcription. It's when we're copying down information from DNA to RNA. We're still staying in the language of nucleotides. We're still in nucleic acid mode, but we're just transcribing. We go to the DNA, we see what the message says, we copy it down in a form called RNA. We also are going to be using ribosomal RNA. Ribosomal RNA, along with ribosomal proteins, are going to make up ribosomes. Now, if you remember from our earlier discussion, ribosomes are our protein factories. They are going to be responsible for copying down the information or reading the information in RNA and then building up a protein based on the information found there. Finally, there's going to be tRNA, or transfer RNA. tRNA transfers the appropriate amino acids to the ribosomes. This process is known as translation. The reason we call it translation is because we are translating from the language of nucleic acids into the language of amino acids. We're going from nucleic acids to proteins. And in order to do this, we're going to need a translation guide or an RNA to amino acid dictionary. The dictionary that we're going to use is called the genetic code. The genetic code is going to be an RNA sequence. The RNA sequence only consists of 4 nucleic acids, while there are 20 different amino acids available. So thus we cannot have a 1-to-1 ratio of nucleic acid encoding for an amino acid. Instead, the genetic code uses codons. Each codon consists of 3 nucleic acids in a row that correspond to one particular amino acid. It's kind of like using letters to symbolize words. In this case, the letters CUU mean the word or mean the amino acid leucine. The letters CCU, on the other hand, mean proline. The letters CAU mean histidine. So what we're going to do is use this genetic code to help us break down or translate DNA into, or translate nucleic acids into amino acids. Now here is a diagram that's provided in your textbook to help you. When you are translating your codons, you're going to start off by finding the first base here. So let's say I have a codon ACG. I'm going to start off by going down the first base. My first letter was A. My second letter was C, and for that I'm going to look across this row at top. So A is the third row, C is the second column, and then my last letter was, I don't even remember what I said. Did I say ACA? I said ACG. ACG is what I said. Down to the fourth row over here on the far side. So ACG is threonine. This is not my favorite chart. It is the one provided to you by the textbook, so please feel comfortable using it. However, there are a lot of much easier to read charts found out there on the internet. I will see if I can include some in our class materials, but if you would like to send me a better chart or send me a chart that you find more useful, that would be worth an additional half extra credit point. So please send me your favorite chart and I will share it with the rest of the class. So that is going to be our table for the genetic code. The last or the remaining sections of this chapter, we're going to be breaking down those two steps, transcription and translation.